The paper that I have decided to review is Requirement for Integration of Signals from Two Distinct Phosphorylation Pathways for Activation of MAP Kinase, written by Neil Anderson, James Mahler, Nicholas Tonks, and Thomas Sturgill, and published in 1990. This paper discusses the protein MAP kinase and its activation. I chose this paper because of my interest in signaling pathways and MAP kinase. First, what is a MAP kinase? Mitogen-activated protein kinase, or MAPK, is one kinase in a family of protein kinases that play a vital role in cell signal transmission. Signals from extracellular mitogens, such as epidermal growth factors, are transmitted to intracellular targets that causes changes in cell proliferation and the cell cycle. These kinases are activated through phosphorylation on specific tyrosine and threonine amino acid residues on the protein. The role of phosphatases. MAP kinase is activated by phosphatase 2A and CD45. Phosphatase 2A is specific for serine and threonine residues. It completely deactivates MAP kinase by dephosphorylating phosphothreonine and not phosphotyrosine. CD45 is a phosphotyrosine-specific phosphatase. It deactivates MAP kinase by removing phosphotyrosine without affecting phosphothreonine. The authors of this paper conducted different experiments to prove that these phosphatases activate MAP kinase and are specific to the amino acid residues on MAP kinase. The first of these experiments was the deactivation of MAP kinase by phosphatases. MAP kinase was purified from 3T3L1 fibroblasts and treated with epidermal growth factors. The purified MAP kinase protein was incubated with either phosphatase 2A or CD45 with protease inhibitors to prevent contamination. Controls use inactivated phosphatases that inhibit 2A. Within 20 to 40 minutes, MAP kinase was almost completely deactivated when incubated with CD45 and phosphatase 2A, while controls showed minimal change in MAP kinase activity. The specificity of CD45 indicates that the phosphotyrosyl sites on MAP kinase are regulatory sites. Another experiment performed was the phosphoamino acid analysis of MAP kinase. MAP kinase is labeled with radioactive phosphatase and treated with phosphatase 2A and CD45. Untreated MAP kinases contained both phosphotyrosine and phosphothreonine. MAP kinases that were treated with phosphatase 2A removed phosphothreonine, and MAP kinases that were treated with CD45 removes phosphotyrosine. Inactive phosphatases showed no change in phosphoamino acid content. The results of this experiment show that MAP kinase needs both residues to be activated or deactivated, and the loss of either would equal the loss of activity for the entire protein. One of the final experiments performed was the simultaneous treatment of MAP kinase with phosphatase 2A and CD45. For this experiment, MAP kinase was labeled with radioactive phosphatase, and then incubated with phosphatase 2A and CD45, with the radioactive phosphatase being monitored for release. The results showed that radioactive phosphatase was completely released, confirming two phosphatases acting on two different phosphorylation sites. All of this evidence comes together to show that MAP kinase must be activated by phosphatase 2A and CD45, and that both of these phosphatases are specific to the phosphotyrosine and phosphothreonine residues found on MAP kinase. This paper helped push the identification of MAP kinase signaling pathways and how MAP kinase is used. Following this paper, more pathways were identified through MAP kinase, such as ERK and P38. Other papers discuss the role of dual specificity kinases and how they regulate MAP kinase by dephosphorylating threonine and tyrosine. Due to this paper discussing the MAP kinase pathway, it was also discovered that dysregulation of the MAP kinase pathway is linked to many diseases such as cancer. This paper resulted in a better understanding of not just MAP kinase, but cell signaling as a whole. It gave us a better understanding of cell signal integration and specificity. It helped us develop drugs that target specific pathway components, and it has driven the advancement of techniques to analyze protein phosphorylation, protein-and-protein interactions, and intracellular signaling dynamics. The hypotheses proposed in this paper are now considered proven fact, and has resulted in MAP kinase being a fundamental protein to understand cell signaling.